Hello and welcome to Lecture 10. In Lecture 10, we're going to look at infectious disease. We're going to look at the human microbiota. So we're really looking at two major situations where microorganisms interact directly with our bodies. And we're going to see a lot of the terminology and concepts that we use when we are discussing infectious disease and the microbiota. And we're going to look at the concepts that we think about. So where we're going to start is with the human microbiota, and it's a form of symbiosis. So symbiosis is a situation where two organisms are living close together for a long period of time. And if you think about it, what is a long period of time? What is a long-term association? If one of the members of this symbiosis is a bacterium that has a maybe 1 hour generation time, a day is a long-term association for a bacterium. So if you get a bacterial infection, that is a form of symbiosis, because for the bacteria, that is a long-term association with your body. Now, if that sounds strange to you, that's because we typically say symbiosis when we mean a very specific type of symbiosis. So there are multiple types of symbiosis. And when we say symbiosis, often what we mean is mutualism, because this is when the host, that's us, and the guest, that's the microorganisms, if we both benefit from that association, that's a mutualism. On the opposite end of the spectrum is parasitism, where we are damaged, but the microorganism, the guest, benefits from it. So that's like a disease is an example of parasitism, where we lose and they win. And there's a whole range of symbioses between these two. In the middle is commensalism, where we don't notice anything. There's no benefit or loss to us on having them, but they gain a benefit. So certainly lots of the microorganisms we interact with don't affect us in any way, but they get a place to live. They might get food. So the tricky thing here is that we don't know the effect most microorganisms have on us. If you look in a gut where we have huge, astronomically huge populations of microorganisms, there are many, many, many different species. And for any given one of them, we don't know the effect it has on us because we don't know what it's doing there. If we're really curious about it, we can figure it out with a lot of experiments. But for most of it, we don't know yet. And so the most common situation, the most common symbiosis between humans and microorganisms, we don't even have a word for it. It's where we don't know the effect on us, but the guest benefits. And so symbiosis has this wide range, and we like to talk about the mutualisms, we like to talk about the parasitisms, but the ones where it's just unknown is the most common. Okay, so let's look at some the related terminology we're going to have to use a lot today. Disease is going to be a situation where our state of health is decreased. So something's wrong with one of our systems. And an infection is where microorganisms are growing inside us or on us. And this can happen without disease. So technically, the bacteria growing in your gut are infecting you. And when I was in graduate school and I used to feed caterpillars bacteria to study what would happen inside their guts, the medical microbiology people I collaborated with would ask me the conditions under which I would infect the insects. And I'd say, what the heck? I'm not hurting them. But they were right, because infection just means the microorganisms are growing inside the host. So as you can imagine, what we're going to talk about in this class a lot is infectious disease. So disease caused as part of an infection. There's a lot of terminology around pathology. So when I hear pathology, I imagine a pathologist looking at microscope slides in the basement of a hospital. But pathology is the study of disease, and there are a lot of path terms around it. So I say pathogen a lot when I'm talking about a microorganism that causes disease. Pathogenesis and pathogenicity describe pathogens and their ability to cause disease. So a lot of people I know research pathogenesis. So they research what it takes for a particular microorganism to cause a disease. There's going to be a lot more terminology later in this set of slides, and I try to organize it, and you'll see the titles are a little bit weird because I try to group it in a way that will be as straightforward as possible. And so if you take a second to understand what I'm doing with the titles later on, when you see weird letters and numbers, that will help you. But for now, let's jump into the human microbiota. Now, microbiota is a word you might not hear much. You might have heard the human microbiome. And when people say microbiome or microbiota, we're talking about the same thing. But specifically, microbiome refers to the genetic content of the microorganisms. And the microbiota just refers to the organisms. So their genes, their proteins, their peptidoglycan, just the whole cell. So I say microbiota because that is what we are really talking about. You're also going to see terms like the resident flora. Flora means plants, and that was what people used to think microorganisms were part of. Don't say normal flora. If you say normal flora, it's clear you haven't taken microbiology for a very long time. Anyway, this is kind of a wild thing to think about, is there are a lot more bacterial cells in a body than there are human cells in a body. It could be a factor of 10 different. It could be a factor of 100 different. You'll see both of those numbers. Part of the problem is that there's a lot of variation between people in how many bacteria they have, and it's hard to count the cells in a human body. No one has a really good count of how many cells make up a human body. We are getting some good estimates, but do you know how many cells make you up? You might have a guess. I don't know. So obviously human cells are bigger than bacterial cells, so we don't care that we have a huge number of bacterial cells because they are microscopic, but they have genes that we don't have, and those genes encode enzymes that we don't have. So their gene products give us metabolic capabilities we would not have otherwise. So they basically make our gut into something that can process foods that we otherwise would never be able to digest. And so they help us extract more nutrition from our food, and they do lots of other things that we are slowly learning about. I have to also mention that there are all these different things in our guts. It's not just bacteria, but fungi and protists. For all of them, there are also viruses in our gut. Every cell on Earth is outnumbered by every virus on Earth. What I should say is that for every cell on Earth, there are 10 virions, roughly. So if you go looking for virions using the tools we have to use, you will find them. So one thing to think about is, where is our microbiota? And there are defined locations where our body lets microorganisms live. But if you find them in a muscle, or if you find them in the dermis, that's usually the sign of a possible disease. So look what I did up here as part of this title, RM1. That stands for Resident Microbiota 1. So as long as you see RM dash a number, we're talking about the normal microbiota. I did this because these slides might not all seem like they go together. You'll see. So here's some terminology to think about. The resident microbiota, RM, are the ones who live in us and on us as part of a symbiosis. The transient ones, that's the opposite of resident, they pass through us, or they land on us and fall off. So you'll find them on you, but you won't find them the next time you look. Whereas the residents will be there both times. And The transient ones are the ones you remove by washing your hands, usually. The transient ones are the ones we can, remove through asepsis and using antiseptics. What we cannot do is get rid of our resident microbiota, typically. They're stuck to us very well. This is, for curiosity, this is a list of the different places on a body where we would expect to see different microbiota. And it tells you some of the members, so some of the genera, some of the species, and comments. It has the curiosity symbol. You can skip this one if you want. I talk mostly about the large intestine because by mass, that's where 99.9% of them are. And it's the most diverse in terms of the number of species you will find. And it's the most active in terms of the metabolism going on there. It's an unimaginably complex community of microorganisms in the gut. And realistically, that is feces. Human feces is 50% bacteria by mass. And that's why it smells bad. It's like bacteria that are aggressively metabolizing different stuff with no oxygen. So they're doing fermentation and they're doing anaerobic respiration. And a lot of times that smells terrible. So your feces basically contains an amazing community of microorganisms. Okay, so how do they get to us in the first place? Well, during birth, typically in the birth canal, the mother inoculates the newborn. And breastfeeding also transmits different microorganisms that help digest breast milk. There are complex sugars in breast milk that humans can't really digest, but the bacteria that colonize the breast allow the child to do it. And those would include like bifidobacterium. And so in a situation where a child is born through cesarean section, a lot of people intentionally expose the baby to normal microbiota later to give them what they would have been exposed to had they been born vaginally. So the colonization happens early, and then once a baby transitions to solid food, that's when they start acquiring an adult-like microbiota in the gut. And there are a lot of ways that our microbiota protect us. It's called, one of the basic and best studied ways is colonization resistance, or the barrier effect. And that's where if we have resident microbiota on us or in us, it makes it less likely for us to get other infectious diseases. So those microorganisms that we have that long-term association with protect us from others. And there are other ways they help us. So they help us, like I said, with undigested food. They protect a lot of our mucous membranes. And so one way to think about it is, what do they do first on our surfaces? What do they do on the outside of our bodies? And one thing they do is they occupy colonization sites. So the normal microbiota have the ability to attach to our external cells really, really tightly, really, really well. And a lot of like disease-causing organisms don't. They also sometimes attack. Some of them will release toxins that will poison other microorganisms. And then in other cases, just the byproducts of them living damages others. So the clearest example of a non-specific antagonism, so I should say, an example of this competition with transient biota, that's on the skin in many cases. That's where we think of that, where you have certain bacteria that live on the skin that protect you from other bacteria, just by occupying space and protein attachment sites. Specific antagonism, we don't normally talk about many examples of this. They exist, but we're not getting into it. Non-specific antagonism is the biggest thing we think about here. And the clear examples of this are going to be like In the gut, removing all the oxygen makes sure aerobic bacteria can't grow in the gut. And so a lot of bacteria that need oxygen will never survive in the large intestine. The vaginal microbiota is a very clear example of this. The lactic acid bacteria in the mucosa of the vagina, they use carbohydrates that we give them, and they ferment them, and their fermentation product is lactic acid. And lactic acid both decreases the pH and acidifies it, but it also acts as a proton uncoupler. That means it shuttles protons across the cell membrane. And so an invading bacterium that wants to grow there tries to set up a proton motive force, and it can't, because the lactic acid destroys their proton motive force. Whereas the lactic acid bacteria, they're adapted to this. It doesn't hurt them. So that is a clear example of microorganisms protecting humans. So I'm going to end this video here, and what we're going to look at next is the situation where the benefits they give us break down and stop working. So what happens when we lose the protective functions of the microorganisms? So I will see you in the next video.